Welcome to this explainer. Today, we're diving straight into the fascinating microscopic machinery of cancer treatments. We're going to see exactly how scientists managed to stop a runaway cellular factory. If you've ever wondered how these life-saving drugs actually do their work down on a molecular level, well, you are absolutely in the right place.
To really understand modern medicine, we have to ask a fundamental question. How do you put the brakes on a biological assembly line that has completely bypassed all of its safety checkpoints? See, in a healthy body, cells divide in a highly regulated way. But cancer cells? They're like a factory where the supervisors have been locked out, the assembly line is running at triple speed, and the stop buttons are literally broken.
When we look at standard cancer treatments, we generally divide them up into two categories. Traditional treatments, like surgery and radiation, those are local. They target a specific physical area of the body. But chemotherapies and these newer targeted therapies are systemic. They travel throughout your entire system, aiming right at the microscopic proteins involved in cell division. They're basically hunting for the gears of that runaway factory.
Now, our literal cellular factory floor is the cell cycle. As you can see, it's divided into distinct phases, G1, S, G2, and M. And each of these phases has its own crucial security checkpoints. These are basically questions the cell has to answer, like, hey, is all the DNA replicated, or is the environment favorable right now, before it can proceed. The problem is, cancer cells figure out exactly how to cheat on these questions.
So here's our map for today's explainer. We'll be looking at 1. The runaway cell cycle. 2. Master controllers. 3. Sabotaging DNA replication. 4. The spindle trap. And finally, 5. Cutting off growth signals. Let's trace the journey of a cell through these phases to see exactly how different classes of drugs sabotage this rogue machinery.
Section 1. The runaway cell cycle and master controllers. Let's dive right into this first part and meet the actual biochemical engines driving this microscopic assembly line.
Before we get lost in the jargon here, let's clearly define our key players. Cyclins are stage-specific proteins that essentially act as the keys. Kinases are the enzymes, specifically cyclin-dependent kinases, or CDKs, and these are the engines. On their own, they're completely inactive, but when they combine, that's what powers the entire cell cycle forward.
Just look at how the cyclin physically binds to the cyclin-dependent kinase. You can see how they lock together, creating a fully active engine. Once this happens, the complex can start phosphorylating other proteins, effectively pushing the cell right into the next production phase. It is the ultimate go signal.
Now, because cancer cells often overexpress these exact CDKs to aggressively bypass natural checkpoints, scientists develop therapeutics to target them. Highly cytotoxic drugs, like flavopiridol, act as broad-spectrum jammers. By inhibiting multiple CDKs all at once, including CDK 1, 2, 4, 6, 7, and 9, they effectively pull the plug on the power supply. This forces the cell to arrest right there in the G1 or G2 phase before it can even divide.
Section 2. S phase, sabotaging DNA replication. Let's move on to the S phase, where the cell copies its DNA, and see how this builds into an opportunity to intentionally throw a wrench right into the biological copy machine.
Think about it. If a cell is going to divide, it absolutely must replicate its DNA. Some therapeutics, like hydroxyurea, shown here, act as antimetabolites, and they are incredibly sneaky. They essentially starve the cell by depleting the actual raw nucleotides, the building blocks that are available for DNA replication. If the factory doesn't have the parts, well, the assembly line stops cold.
Other drugs target entirely different machinery during this phase, specifically topoisomerases. You can think of topoisomerases as the essential detanglers of your DNA. They cut and reseal the strands so they don't knot up while being copied. Drugs like camptothecin act as poisons that freeze the enzyme immediately after it makes a cut. Meanwhile, doxorubicin poisons a different topoisomerase, topoisomerase II, leading to massive, catastrophic DNA double-strand breaks.
Take a look at the complex chemical structure of doxorubicin. It's a powerful S-phase inhibitor. By interfering with that topoisomerase, it ensures the unwinding gears of the DNA are completely paralyzed and heavily damaged. The cell is just left with these shattered DNA blueprints, making it totally impossible to safely proceed with division.
So the really crucial point here is how the cell actually senses this massive damage. Our cells aren't blind, right? They have highly sophisticated alarm systems. The ATM and ATR proteins act as the biological smoke detectors. They detect these double-strand breaks or stalled replication forks. And once they're triggered, they phosphorylate key downstream proteins, including the famous p53 tumor suppressor protein, which basically yanks the emergency brakes on the whole operation.
Following this cascade, you can see exactly how X-rays or chemotherapy drugs cause that initial break. This activates those ATM and ATR kinases, which then stabilize the active p53. p53 then goes straight to the DNA to bind to the regulatory region of the p21 gene. And the result? The production of an inhibitor protein that shuts down the CDK engines entirely. The cell is literally forced to halt.
Section 3. M phase, the spindle trap. Now we enter the M phase, or mitosis. This is where the cell has finished copying everything and tries to physically divide into two, running right into the strict spindle assembly checkpoint.
Notice the stark difference between pro-metaphase on the left and metaphase on the right. For a cell to divide successfully, every single chromosome must align perfectly at the cell's equator, held under strict tension by these microtubule cables. Only when this exact tension is sensed across the kinetochores is the cell actually allowed to split. It's basically a microscopic game of tug-of-war.
And this is where nature provides one of our most fascinating weapons. Incredible compounds called taxanes, which were originally derived from the bark of the yew tree, are introduced. These drugs physically freeze this division machinery by inhibiting microtubule depolymerization. They basically glue the ropes in place, meaning the tension is never quite right. This activates a permanent tension alarm, permanently trapping the cell in mitosis.
Section 4. Cutting off growth signals. Finally, we're going to step outside the nucleus entirely to see how cancer cells hijack external communication lines and how targeted therapies act like biological wire cutters to silence them.
You see, cells don't just divide randomly. They need a signal from the outside world telling them to grow. Tyrosine kinases act like communication antennas on the cell surface for these exact growth signals. But in cancer, receptors like EGFR or HER2 become mutated. They get stuck in the on position and just constantly scream at the cell to divide and multiply, even when there are absolutely no actual growth signals present.
We can intercept these rogue signals in two incredibly ingenious ways. The first is with small molecules, like the revolutionary drug Gleevec. Because they're tiny, they sneak right inside the cell to physically block the ATP binding site of the kinase, effectively cutting off its power. The second way is by using large monoclonal antibodies, like Herceptin. These are too big to go inside, so they attack the receptors directly from the outside, binding to them and causing the cell to pull the receptors inward and dismantle them, a process known as downregulation.
Interestingly enough, these rogue, mutated kinases are notoriously unstable, and they're often kept intact by a stress-sensing chaperone protein called HSP90. Think of HSP90 as a bodyguard, but experimental drugs like geldinomycin can actually block this chaperone. And without its bodyguard, the dangerous mutated kinase just falls apart and is sent straight to the proteasome, which is basically the cellular trash can, for degradation.
So we've thrown everything at it. We've stopped the CDK engines, broken the DNA replication machines, frozen the dividing spindles, and cut the communication antennas. But what is the ultimate intended fate of this stalled, broken factory? Do we just leave it there?
Well, the ultimate goal of all these precise therapeutics is not just to pause the cell, but to force it past the point of no return toward apoptosis. Apoptosis is regulated, programmed cell death. Rather than just bursting and causing massive inflammation, the cell neatly dismantles itself. This is marked by a characteristic blebbing or bubbling of its cellular material, safely clearing the threat from the body altogether.
Which leaves us wondering, as our biological wire cutters and jammers become even more incredibly precise, bypassing healthy cells to target only the rogue ones, how close are we to perfectly controlling the off switch of cancer? The molecular assembly line is endlessly complex, but as we've seen today, our tools to dismantle it are becoming absolute masterpieces of science. Thanks for joining me on this explainer, and keep asking the big questions.